Hey, thanks everybody. Good morning. It's terrific to be here. And thank you to the chairs of the meeting for the invitation. It's quite breathtaking to see the uptake of genomics in personalised medicine. I think I was just out of the country for 3 weeks, and I think there were significant developments even while I was gone. So that's rapid progress. I guess this is a new slide for me. This is the learning objectives for the talk. The concept of individualized medicine based on complete understanding of the nuts and bolts of how we are put together is not new. And the idea that we, through the Genome Project and other exercises in molecular biology research, the idea that we can completely describe all the parts of cells, and then through the description we can then build this understanding and ultimately have sufficient understanding that we can do manipulation and treatment of biological systems is not new to any of us. It's a basic concept. And of course we want to understand things not like Swiss watches, we want to understand individual humans. But what I think is new for us and is being reiterated right now on the shop floor is the idea that this is not a unidirectional process. We don't simply go and understand biological systems through applying genomics. We, in fact, as we apply it, we learn more, and then we reinforce our models and build this cycle of understanding. And that genomic medicine is, in fact, now playing a role in this cycle. It's not just the recipient of the knowledge from these basic studies, it's really embedded in this cycle. Now, this is all possible, of course, because we couple genomics to the basic principles of genetics. And we undergo what we might call biological or medical genetic determinism, the idea that if we understand the gene, we can understand the phenotype. And of course, you all know, I'm sure, that kind of an idea falls into at least two arenas. One is the simple one, where we have simple genetic problems through single monogenic diseases, and then the complicated one, which involves most of the diseases which ultimately will kill us, things like heart disease or cancer or behavioural disorders. Now, since we want to understand these biological systems in this intricate way, and we want to use genetics, it would be very nice, of course, if we could have not a dichotomized genetic model, but if we could have more of a continuum between them. So this idea of connecting simple problems in genetics and the idea of Mendelian disease with complicated problems in genetics and common disease is not actually new, of course. In fact, it really came to the fore more than 20 years ago with the discovery of the breast cancer gene, where initially we had hoped that if we could understand that 1% of individuals with heritable breast cancer and find the genes that were driving that phenomenon, we could study those genes more and understand the rest of the sporadic cases of breast cancer. Of course, that was not realized initially, but I think you could even say now that we are building that story now so that some of those early lofty aims, in fact, are being realized. Nevertheless, we are back doing this more with full force now in 2014 because of the dramatic changes in technology. Now, here's a fairly Houston-centric view of how those technologies have evolved and how the applications of clinical genomics have started to get to the point where we can reciprocate and understand more biology through their application. After the initial human genome sequence was completed by the International Consortium, there was a lull of about 10 years, and then we embarked upon a number of projects we'll call individual genome projects. And the first of those was the Watson project, and we were fortunate to be able to perform that project in collaboration with 454, a commercial entity. So basically, we sequenced James Watson, who became the first diploid individual to be sequenced, and then analysed his genome and discovered that actually we had more individual private variation than what we had first expected. Now, that's something we all take for granted today with the amount of data out there, but at that time we were quite surprised to see the number of genic changes, several hundred, that existed between Watson and then the very soon after Ventnor sequence. Several years later, we were able to sequence one of my colleagues, James Lupski. Now, Jim is known to some of you at least. He's an expert in a disease called Charcot-Marie-Tooth syndrome. Charcot-Marie-Tooth, or CMT, is a heritable neuropathy, peripheral neuropathy, and Jim himself suffers from the disorder, as well as being an expert in the disease. Back before the molecular studies, this was thought to be one syndrome. It's now known to involve at least 40 different genes. There are 40 different possible causes. And so Jim had found many of those other causes, but never found the cause in his own family. And with these new methods, we descended upon Jim and bled him. And in a few weeks, and for less than $50,000, we discovered the locus that's actually causing the disorder in his family. Of course, that's not an actionable finding in the sense of having a cure, but it was actionable in the sense of changing his clinical prognosis and also in driving some of his research work. A little later, we used the same whole genome methods to sequence some twins who came to our attention, the Beery family twins, and I'd invite you all to go to the Beery family website, as the mother is an impassioned advocate of genomics in medicine. Through the sequencing, we were able to determine that the movement disorder in the children was not due to a defect directly in the dopamine pathway, which was their previous diagnosis, and that the symptoms they presented with, which were quite severe, were in fact due to a cofactor that affects both the dopamine-dependent pathway and the serotonin-dependent pathway. So in fact, a few supplements that provided the substituted for the deficiency in the serotonin pathway completely ameliorated their symptoms, and they went from children with near-fatal breathing disorders to children who actually won athletic championships. So it was quite a dramatic poster child for the actionability of data that you can find in this scenario. So this series of whole genome projects led us to the point where we thought we should be more aggressive in bringing these methods into the clinic. They were all driven by whole genome sequencing, but about the time of the Beery study, we developed these capture methods where you're able to pull exomes themselves out of the genome and just sequence the 1% that is interpretable at this time. And of course, there's a debate to be had about what you're missing out there, but the fact is, still to this day, virtually everything we know about the genome is contained within exons. Now, on that note, I'm not going to launch into any technical deep discussions, but I do want to iterate that in the context of this debate about genomes versus exomes, the exome methods and the yield of those and the part of the genome they cover is improving constantly. We've been working to increment the methods. They're getting faster. We've gone from an 18-day down to a 4-day protocol, very recently a 1-day protocol. And we have what we call a panel killer program, which aims, instead of following the philosophy that if you just examine a very small number of genes, you can do them with much more sequence depth, we can, if we tune our exome reagents correctly, get very good cover of all the coding regions. And you'll see in the very bottom right of that slide, there's a red and a blue part of the histogram. You'll see the increments, small increments, but very significant over genome space, of the amount of coverage we are getting from these methods. So people always ask me, when are the exome methods going to go away and be replaced by whole genomes? And I say, well, the moment they're about perfect, which is usually what happens with these technologies. Now, this is the only other technical slide I'll give you. It's a sort of snapshot of where we're at. Most of you know that there's a new sequencing platform available. If you spend $10 million on 10 machines, you can get what is, at its base cost, near a $1,000 whole genome. And you should ask, and many ask then, well, doesn't this completely obliterate the exome genome discussion? And in fact, the vendor has constructed the sale of these machines with a licensing agreement that insists that you only at this time do whole genomes. You cannot do exomes or other types of substrates on these machines. We're not sure where that agreement will go, where it will unfold. But of course, the counter then to this point is if you can do a $1,000 genome on one of these machines, you can probably do an $80 exome on one of these machines, $80 to $100. Can you imagine the impact of these technologies if for $80 you could get a whole exome worth of data? That's really below X-ray costs. So in our institution and elsewhere, many other places, these methods have very dramatically driven the rate of discovery of new monogenic disorders. And here's just a snapshot of individual papers that describe exome discovery of new genes. But I'm sure any of you who picked up the American Journal of Human Genetics or other similar journals will see that the journals are populated very densely by these kinds of new discoveries at the moment. So what's happened, a phenomenon that has accompanied this is the shift of single investigators who go out and take full responsibility for soup to nuts discovery of these new single genes is a more institutionalized and a more industrialized process. And part of this has been driven by a National Institutes of Health program that established the Centers for Mendelian Genomics. We have one of those in Houston. It's a collaboration between the Genome Center and Jim Lupsky, who is the PI of that. And the idea is simply to accelerate the discovery of all these single genes. Now remember, there's 20,000 genes. There were, at the beginning of this program, about 2,500 of those that had been linked to phenotypes. The OMIM, the Mendelian Catalogue online, lists about 10,000 disorders, of which it was thought about 8,000 at least should be tractable, that we could discover a phenotype associated with the disease. It may be that there are even more, especially if you consider those that are lethal. Now, from the outset, it was realised that there would be a paucity of samples to feed into this discovery effort. That is that over the many years, using slower technologies, the reservoirs of interesting families that clinical geneticists had accumulated in their refrigerators had largely been depleted. There would be some more of those, but there would be some need to accelerate sample capture. So the solution from Houston has been to have a series of genetics fellows who travel out to their home countries as well as, and retrieve samples and send them back, as well as to use the general network of clinical geneticists, which is not such a large community, and many are known to each other. There's also been an approach attempting to do this by having a website where individuals will come with their samples and deposit them to the program, but that's been relatively unsuccessful, an interesting sociological exercise. So we have now literally thousands of samples that have been sent into the central site for sequence analysis and for discovery of these Mendelian single causative genes. And the numbers are very encouraging. This slide is actually a little out of date. There's probably about 300 novel genes that have been discovered in the Houston program and a couple of hundred more across the whole venture. In some cases, these are truly novel genes in that nobody thought they would be involved in a phenotype, and now they are linked for the first time. In other cases, it's what we call phenotypic expansion. The clinical diagnosis was ambiguous or wrong or just not known, but when we have the molecular data, we see that there's a different set of phenotypes than traditionally associated with that locus. So we've linked the gene to a known disorder, but the phenotypic description is now more broad than it used to be. So the point here is that there are hundreds of these being discovered, and that's being driven by these sample collections that are done ad hoc around the world. Now, although we're now speaking of hundreds, it turns out that if you want to apply the criteria of true discovery to these genes, it can still be very hard work of the single investigator type that we're more used to really get conclusive proof. For example, here's one single allele that was discovered in two different families, actually by two different Mendelian centers, which causes brain malformations. It's a gene called CLIP1. It's low frequency, so we didn't have the ability to see it in many families in order to validate its presence by statistics. But we were fortunate that there was a mouse deficiency model, and when that was exploited by postdocs, we discovered that indeed the phenotype in the mouse matches the phenotype, or correlates with the phenotype in the human, and we crossed that barrier for proof that one needs to publish this locus as a cause of a new phenomenon. So this is good on the one hand. It's one gene, it's one allele and two families, and it's two cell papers and a couple of postdocs doing months and months and months of work collaborating with an international consortium. What's not good is if you try to imagine scaling that across the whole demand to discover all of the rest of the Mendelian genes. So is this an isolated observation? No. If you look at many other examples, and I've just put three others in three of the panels on here on this slide, we find the discovery of these alleles when we go into cellular assays or animal assays in mice or in zebrafish. It's months and months and months of individual investigator work to establish this causative, this notion of proof that the locus is truly driving the phenomenon. It's just not scalable. So the question immediately becomes, of course, well, how can we scale this? And the answer is the statistics and the segregation statistics of the phenomenon. So, for example, in a recessive case, you might find a single family with one gene that segregates nicely with two, looks like they're causative alleles associated with the phenotype. If you can establish that there are other causative alleles in other families in the same gene, in multiple other families, then you can quickly get to this level of proof without any functional data. Now, it's a little more difficult in cases of dominant disorders, but there's a phenomenon that's now been recognised, long known, but now recognised for its frequency and importance, of the occurrence of de novo mutations driving these single gene defects. Now, we each have a couple of hundred de novo mutations in us per individual as we are formed. And that leads to somewhere between 1 and 2, usually, genic de novo mutations per individual. And the basic concept of using de novo mutations to discover new disorders is that if you find a couple of individuals, 2 or 3 or hopefully 4, who have the same gene, similar phenotypes and the same gene with de novo mutations that remove the function of the gene, then you've got statistical proof that that mutation is causative.
Dr. Gibbs, I'm very sorry to interrupt you. Everybody, we have a fire in the kitchen. It's okay. We're going to stay put, but we think we're going to lose power for a moment.
Oh.
So if everybody can just hang tight with us, we thought you might lose lights and slides and microphone.
Would you want me to press on to that, or do you want to?
Yeah, let me just check, but I wanted everybody to be aware that this might happen momentarily. So let me just give a quick status check.
Thanks, that's a first. Wow, thanks everybody. Thanks to the crew out there too. They were working pretty feverishly, I think, while we all took a break. So let me recap very briefly. We've got a lot of good sequencing machines, a lot of good technology, a rate of discovery that's impressive by old standards, but a limitation of samples to drive single gene discovery. So what are we going to do? Well, right at that time, in Houston, we decided to port over some of these methods into a clinical setting, and we built the whole genome laboratory. And the idea here is that instead of doing a research-style exome and discovery, we would simply get samples in for routine diagnosis of pediatric cases with a suspected genetic condition. This is actually a fairly difficult thing to do for any group. It involves a lot of different moving parts, and I often get asked by other groups, how do you put one of these things together? And the answer is simply, it takes a village. Oops, I'm sorry, there we go. So we did that in 2011. So we conceived it in about June of 2011, launched it in November. And this is the histogram showing the number of samples per month that have been through the facility. It's around 250 per month now, or it's reached 250, I should say. So that's actually a lot of work. It's like 250 single genome projects. And the rate of solution of the cases that come in is about 25%. That seems to be a standard now across different areas. There's a total of about 5,000 samples that have come in the door. About 4,000 reports have gone out. Now the reports that, I should say that we've built new algorithms like some of you have to do the genetic interpretation with the hope that one day we can just get the sequence and push a button and have an answer. But we're still actually very highly dependent on the community of expert clinical geneticists. And you see a bunch of them here at a monthly sign-out. We've been putting these monthly sign-outs on the internet. So if you want to go and click on the website there, you can actually see videos of some of the sign-outs of the cases. The report is sort of an uninspiring looking document, but the content is, of course, what's important. The main report that's a default is a report of the deleterious mutations that are related to the disease phenotype, variants of unknown significance that are related to those genes that are known to cause Mendelian diseases are reported, and then actionable incidental findings are also reported. Then there's a series of other things, including an optional non-actionable incidental findings, and perhaps that's a point we'll discuss after. If you want, you can get a full-blown report that has all of the molecular data, but many don't want that. Just parenthetically, since I'm here with Dr. Winchelbaum, and we've been involved with him in the PGRN program, I'll mention that there are some pharmacogenomic markers that are reported, but not all of them. And of course, there are increments to be made in that. And we've been trying to develop PacBio methods, Pacific Biosystems long sequencing methods, to better report pharmacogenomic markers. My genome report came to me, and I was intrigued to find I have a carrier for 2 different genes, of which the autosomal recessive state gives blindness and acute hearing problems. And I'll come back to that because I do have something of a hearing issue, and I do have then a mild carrier burden in those disorders. So this is a theme that I want to come back to in just a moment. But the basic message I can give you now is that this clinical service, which is now proceeding at this pace, which is now larger than the research discovery unit for single gene disorders, is giving us all of the kind of information that we were getting from the research program. So within the clinical program, we're seeing phenomenon like uniparental disomy where there's vast amounts of homozygosity caused by chromosome segregation issues being abnormal. There is cases where the complex phenotype has been the result of two different independent single gene disorders in the same person. And the phenotypes in those cases can generally be retrospectively resolved into those two genetic components, but simply you're talking about a child who's basically got two different single genetic diseases. And there were 4 of those in the first 250, and that statistic has been borne out with the thousands more that we have. Now, I'm making it sound like we're just doing research on this set, but actually we can't do that. There's a big wall up, of course, between the patients and the clinical test consent that they sign and our normal research program, which has a much more detailed consent program. So much of our work at this time is in taking the cases that are not solved from the clinical cohort and bringing them over that wall by reconsenting them. In the instances we can do that, the results are really quite stunning. And back to this issue of the power of de novo mutations to define a disease, here's an example where we had initially had a child who was referred to our research group that had tremendous debilitation. The child passed away at around 6 months. We had the trio of sequence. We had a new mutation in a gene that was not previously known to be associated with the disease. The gene is called ASXL3. Now, ASXL1, related to ASXL3, they're both transcription factors in the same family, is well known in a different single gene disorder. And we were able, through networking into clinical geneticists and ultimately looking in our own database, to find 3 additional cases of ASXL3 de novo mutations with similar phenotypes that retrospectively, once we had the molecular data, we could see these phenotypes are indeed related. And this, in fact, defines then a whole new syndrome based upon ASXL3 deficiency. There are 4 cases there. Since then, there's been another handful that have been reported. Here's another story. Different children, different gene, exactly the same mode of discovery. Here, this is actually the child in the top left of the panel is my grandniece, and I persuaded the family to come into our clinic, and we indeed found a new mutation in a gene that looks suspicious. In our own database, we found 3 more, overlapping phenotypes, hypertonia, sleep apnea, speech delay, and we defined a whole new syndrome based upon that. Now we have an additional 3 children with the disorder. And here's yet a third story where I'll tell you exactly the same thing that Seema Lalani has driven. A new gene. The gene is actually in a region, in this case, that was known to be part of a microdeletion syndrome, but now the de novo mutations have narrowed down the field to identify a single gene. So this is the power of de novo mutations to define these new disorders, if you can get the consents from the families and allow them to be brought into the research arena. A good question to ask is how frequent are these disorders if we're seeing them in Houston at a frequency of 2 or 3 or 4 in a couple of thousand. And for that, to do that calculation, you have to make some guesstimates about what fraction of the live birth population we're catching and about what fraction of the, what window, how many years of age the children are that come in. But in each case, we can say for these disorders, the number's probably somewhere between 500 and 5,000 children with each of these disorders internationally. That's a very broad margin. But these things are rare individually, but when you take the size of the whole human population, it adds up to a lot of children. Why is that important? Well, it's important not just because it's the health burden, it's because there are many more instances where we can look at these individual genes in different contexts and decide what the mitigating factors might be for the full expression of the phenotype. Now, another good question to ask is since we have now these several thousand cases in the clinical database, if we could mine them freely without any constraint, what would we find? And what this slide basically tells you is there's probably another 100 to 200 genes which could be added into the Mendelian catalogue simply through that unrestricted mining. Because there are many, many examples of individuals there that have the same gene mutated as in multiple other individuals and some overlap of phenotype. So we await getting those samples fully consented to make these discoveries. Now, this is all great. This basically what I've told you since our break is that we've got a clinical enterprise which is bringing us a lot more samples in, is further driving our ability to make Mendelian discovery. These are all complicated Mendelian stories, but saying they're complex and complicated Mendelian stories does not mean that they're instantly in the arena of being related to common disease. Before the break, I showed you a slide about how can we connect Mendelian disease to common disease. And so the question is, how do we do that? And how do we build what's in the middle of this slide oligogenic models, which would be those models of genetic structure of disease where individual genes contribute alleles together, somehow interactively, to lead to the disorder. Well, we're starting to get the kind of data we need that really allow us to do that. And the data speak to the effect of carrier burden in different syndromic situations. So the example I'll show you is in Charcot-Marie Tooth. I spoke about Jim Lupsky's case. That's one of those situations where there are Mendelianizable forms that are in families, but there are many sporadic cases that are just not solved. And what Jim and his student and the Genome Center have done is to go in and study many of those cases that were not previously solved. And the bottom line for that study is, we found a couple of genes that were missed. The bottom line, though, is that many of the cases do not have simple Mendelian stories that explain the Charcot-Marie tooth, but if you look at all genes that are ever known to cause a neuropathy, they carry an excessive burden of heterozygous mutations in that subset of genes when you compare them to the best controls that you can find. And so basically the blue versus the red histogram here shows we're seeing more carrier burden in these Charcot-Marie cases, Charcot-Marie tooth cases, than we are in the controls. Now, we don't have sufficient numbers to do this genome-wide. We have to constrain our study to the 400 genes that are involved in neuropathies. Nevertheless, if we do that, we see the statistically significant effects, and we look forward to having enough samples to do it genome-wide. If we take those loci and work with our collaborators, Nico Katsanis, and that's Claudia Gonzaga, who was Jim's student and moved to Duke, who's now at Regeneron, and we build zebrafish models with different combinations of those alleles and look at the zebrafish, we see indeed that there is phenotypic evidence for interaction between the carrier states that speak to the notion that these loci could be together driving the end phenotype. And if we map those genes across, here's across a peripheral nerve model, but also in mitochondria, and the closed circles here are examples of individuals with the genes that are distinguished by colour and for which a Mendelianizable form. The open circles are ones where there's a carrier burden donated by that locus into our model. We see that they all fit around the CMT space. So the bottom line of what I'm showing you here is that we're on the beginning now, the beginning state of having sufficient sample numbers for syndromic conditions where individuals who don't have simple Mendelian stories do have a carrier burden in a subset of genes that can be related to the phenotype. And others will present to you data on intestinal muscle disorders and on bone in other studies from other individuals. So the conclusion of what I said before the break and now is that we're actually doing clinical translation of genome sequencing quite routinely now in genetics up to this burden of 5,000 samples. And our discovery now is actually becoming dependent upon that source of samples. It's great because the insurance companies are paying for the first pass and for the clear answers. When they're not answered, we move the samples over to the research space and we bring the data with them. And so as we accumulate more samples and we do more of the single locus discovery, we'll also, we believe, be able to build more robustly these oligogenic models that'll bridge Mendelian disorders over to models for complex disease. Now, that's all the substance of my talk here. I just want to speculate for one moment, though, on what this all means for the future. And by future, I like to talk about what I call armchair futurism. And we all know the stories of who's ever going to need a cell phone and who's going to need more than 64K of memory in a computer. To predict the future in a non-linear way is actually, it requires you take a sort of out-of-the-box view of things. And I think the thing that's really most striking to us in Houston is shown here in my very last slide. I'll skip the arguments getting there, but here. We essentially right now are somewhere between that blue circle on the left of the slide, the oval, and the one in the middle. We're doing population disease discovery. We're building our models of genetics here in the laboratory. And we're starting to put those things into the medical record. That's the red circle. And to some extent out in the public, into the social arena. But as the time and technology passes, and it's passing so very much more quickly now, that red circle is growing and growing, so we're seeing more of that. In fact, it's becoming immense. So I think that the futuristic prediction is that many of us here are, if we want to do a genetic study, the first thing we think of is going out to find the individuals with a disorder that we might want to study, or with a set of phenotypes that we might want to study, and we ask how can we bring those people into our research arena. In the very near future, I think many of us will be more naively mining the clinical records, with the appropriate consents, of course, and appropriate structures, naively mining the clinical records to drive our research discovery. And the old enterprise of sending your postdocs to other countries to drive around to collect samples will be very much become a thing of the past. So that's my grand conclusion there, that we'll be entering into this futuristic world. Now, none of this, as I said, it takes a village. This is done by much work by individuals in the Human Genome Sequencing Center in Houston and the Department of Genetics that originally started the diagnostic labs, but with whom we've been working intensely now for about 5 years. And I want to especially acknowledge Eric Bowinkle, the co-director of the labs, and Donna Musney down there, who's the operational director of the Genome Center for more than 25 years now. And many of the other individuals there, I think some you will know through the community. And I want to also thank Dick Wenchelbaum for his collaborations and leadership in the PGRN effort. And I've got to thank you all for sitting through this mosaic presentation. And I think we still have a few minutes left for discussion.
Well, thank you, Doctor, for being so flexible with us this morning. And thanks to everybody for returning. In the meantime, folks have several questions for you. So we're going to try to tick through several of these. You just flew in, but the past couple days, cost has continued to be a part of our discussions here and the concept of value in healthcare. And so not surprisingly, you've got a couple of cost questions. This person asks one I was going to ask. Does it really only cost $1,000 to sequence a genome? And are there any hidden costs?
So no, at the moment, it costs about twice that for us in Houston in a research environment to go from a supplied DNA sample to a sequence with a cursory analysis, the list of variants. The $1,000 is a very trimmed version of the core costs in producing that. I mean, the difference though, I think to some that's a disappointment. To us, to know that it once cost $3 billion, it's really quite stunning to see that.
It is quite stunning, and I wonder, as that cost continues to come down, what might be the implications? I mean, obviously from a research perspective, it's great, but are you anticipating that more just sort of general members of the public are going to say, hey, I want to get this done? And are there other implications to that?
Thanks. That's, of course, an important question, but it's a very difficult one to answer unless you're an expert, I think, in the propagation of health technologies and the history of those, because we certainly see and have discussions where the notion that the service costing $5,000 is prohibitive, whereas at $100 it would not be. Then in other arenas we have discussions where it costs $8,000 for an MRI. And so, if this is going to be critical for guiding some treatment, why on earth wouldn't you want it? And then we see other technologies that may not necessarily be quite the same qualitative type of health technologies, but get propagated with amazing speed because of their clinical demand. And then of course there are triggers like the insurance companies allowing the payments. If these get written into standard of care documents where they're obliged to have them, and of course litigation. If somebody needs to have a test because they don't want to be told they didn't deliver that test in a courtroom.
Right. Right. Another question here, how is the new technology helping us better understand common disorders such as heart disease and diabetes?
Well, there are sort of two pathways to, or two definitions of understanding or of successful outcome. Understanding the nuts and bolts of these disorders is critical at some point, and for sure the technologies are driving deeper analysis of these large cohorts and case control studies that allow us to really look deeply into the genetic signatures of the components that are driving the outcome of those disorders. So we're still making discoveries, and these discoveries have been accelerated by those. Of course, the issue that many get to is what is the value of applying these technologies for the, for prediction of heart disorders or of other common, any common disorder that you might go to your doctor for as an adult. And the truth is that there is minimal impact so far. I mean, of course, your weight and your smoking history and your exercise is much more important for your future in diabetes than what an annual family history, of course,
yes,
then a naive DNA test can be at this time. But that sounds a little pessimistic, and I still think we can carry the faith and have the optimism that we can have greater informativeness from our genetic signatures, even for those disorders.
And you are not the first person at this conference to remind everyone of the importance of family history and how valuable that information is.
Well, I hope I'm not the first one also to remind everyone here that there are many people who are orphans. There are many people who only have fractional knowledge of their family history, or for which their family have not been exposed to the same environment, so they may simply not have expressed the same conditions.
Exactly. Wondering how patients with new disorders are being linked together.
I think it's very ad hoc at the moment. I mean, in a dream world, we would all pull out our genetic data in a safe way that doesn't compromise any of the privacy and other needs of the individuals, and yet would allow full research interaction so that we can make these discoveries much more quickly. But there is really strong and healthy pressure to do that, and everyone is responding. There are different groups that have come together in order to facilitate that. I think one shouldn't underestimate the value or understate the value of what the clinical genetics research community has been able to achieve in its current ad hoc fashion, though. I mean, if we look at the discovery curve, it's going up, and it's being driven a lot by data sharing. And of course, we want it to be facile and free and done in a way that is semi-automated at least. But we're headed that way.
Another one along the same lines, but asking what needs to happen to make genome sequencing more affordable so that everyone can have access to this technology?
Well, I think the role of the vendor in this can't be overstated. Also, the need for competition from different vendors, of course. I mean, right now, the field is dominated by Illumina, who've done a magnificent job of operationalizing this basic technology and delivering it to individual users. And I think that the volume of the activity will hopefully continue to drive their enterprise and make it more cost competitive. But I think we've got to be realistic in a competitive industrial capitalist society. We motivate groups to carry out their obligations to their shareholders. So it'll be pressure through other industrial groups and through other alternate approaches to understanding genetics that will ultimately win out here.
So not necessarily just putting all of our eggs in that one basket, obviously.
That's right, yeah. And I know, I think we can, right now we have this view that in 10 years everybody will be sequenced all the time. We shouldn't be too surprised if some other technology that better captures not DNA variation, but the products of DNA variation, the things we discover through our analysis of DNA variation, actually becomes a more frequently used technology and approach. So, the world changes dramatically, so that's why we should be nonlinear futurists. We shouldn't just take everything we do today and double it or triple it or multiply it 10 times. Things can really change and turn around completely.
Right. Curious, why the pre-publication data from the studies has not been shared more readily?
Well, a lot of the issues are to do with the patient privacy. I mean, and research privacy rules. But then there's a set of issues that are to do with the scientists who reasonably, having studied a phenomenon for many years, would like to enjoy the completion of that study ahead of publication and then full release of all the data. So the question is, well, why not, like the Genome Project did, just put the data out there for everybody to see straight away? And I personally think there's a healthy trend towards that, that for every story where someone says, well, if we just had the data now, then we would have more rapid discovery, there are well-concerted sharing efforts that are bringing these discoveries to fruit. We're seeing, as I said before, we're seeing the growth of the curve, and we are improving the data sharing. I think the constraint on allowing everybody to put all their data out like a Twitter feed for all clinical phenotypic data, I think is not necessarily restraining us as much as some might say.
Okay. Another one from our audience. Other than the $10 million cost for 10 machines, what is required to accomplish sequencing a genome?
Well, the $10 million is kind of the down payment. And then there is, of course, samples need to be brought to this activity, and samples.
How are they typically acquired?
Well, the best is blood, of course, where you get a rich source of DNA, and you're able to extract a lot by fairly standardized procedures, but more less invasive procedures are possible. But the samples need to be of good integrity and need to be organized and managed and then converted into the sequenceable material. So all those steps have individuals who are quite expert that have to be engaged. Then there's the sequencing process, the digitization of the information. Then there's the analysis. And the analysis, of course, is reasonably standardized now. And there are a couple of different alternative pathways that can be used with different tensions between their precision and their false negative, false positive rates, all of these things. So there's an analysis activity that requires experts, which costs money, and for which individuals have to think through exactly the reason for doing the study to decide which pathway to take. And then there's physical storage of information and physical computing of information, which is becoming a significant expense. So again, it's a multi-component process.
Final question for you, and this is, do you envision a day, and if so, when, might whole genome sequencing replace whole exome sequencing?
Yes, I think it will come, but I think that our enthusiasm for it is more bounded by a desire to know things that we don't yet know than necessarily a critical examination of the cost and yield from what we can do. And I think there's an expression that goes something like the absence of knowledge is not knowledge of absence. And so, there's stuff out there that's outside the exons, that's for sure, and we want to find it, and we want to catalogue it, and we want to understand it. But whether or not in a test for a health condition, or even in a discovery mode, there is current ultimate value in having that information versus doing a lot more samples, 10 times more samples, with the more restricted data set, I think is the critical question.
Great. I want us to give a very special thank you to Dr. Gibbs for his patience today and flexibility.
Thank you so much.